Hello, and welcome to the 7th Annual AAKP Global Summit on Kidney Disease Innovations, themed The Decade of the Kidney at Midpoint. Opportunities and barriers to greater innovation and freedom from disease and disability. This annual event is proudly hosted by the American Association of Kidney Patients, AAKP, in collaboration with our allied partners at the George Washington University School of Medicine and Health Sciences. My name is Suzanne Ruff, and I am honored to have served as AAKP former Director, Board of Director, and currently as a National Ambassador from North Carolina. My father was a member of AAKP's board in the early years and taught me how to advocate for patients. I am also a living kidney donor, known as the Reluctant Donor, the title of my book, about how kidney disease has ravaged my family. Before I was born, my grandmother died of renal failure in the 1940s when there was no medicine, no treatment, nothing doctors could do. My mother was a teenager and the youngest of my grandmother's 6 children. In the 1960s, 5 of the 6 children, including my mother, learned they inherited the gene that causes polycystic kidney disease. Mom watched as her older brothers and sisters battled the disease and when there was a waiting list for dialysis. My father decided to have us tested with the now archaic testing available then. All but me inherited the disease. My father was devastated, but found his strength and threw himself into fighting for kidney patients, using his skills and many contacts to help families like ours. His biggest claim to fame was a telephone call that resulted in the first federal funding for the disease. All of us can do something, he often said. More than 25 of my family members have inherited the gene that causes the disease. There have been 10 kidney transplants and 1 liver transplant among my family. 6 deceased donors blessed my family with their gift of life. I was the first living kidney donor, and now we have 5 living kidney donors in the family. Over 20 years ago, when I became a living donor, there was little information for me to research. It was long before social media and before resources like this, the global, emphasis the word global, summit. This is mind-boggling source of information for the layperson to educate themselves, to mingle with doctors, researchers, and experts in the field. I have cried buckets of tears over the 2 words, kidney disease. I have buried many of the people I love most because of it, and I truly understand the toll of watching a loved one suffer from kidney failure. But my family's examples of courage despite pain, faith despite suffering, and good humor through tough times helped me find the courage to go into that hospital and becoming a living kidney donor all those years ago. The advances made since the 40s until now, especially this time, the decade of the kidney at midpoint, gives us hope that through advocacy, awareness, and support, we can help each other to make a difference. Since 2019, the Global Summit has served the global kidney community as the largest patient-led kidney disease innovation summit in the world. This year's Global Summit marks an important milestone because it is the midpoint of the AAKP-led Decade of the Kidney. This global initiative has been mirrored across the world by kidney advocates, medical allies, and government officials, and among people of goodwill, committed to a return of true patient-centered medicine. Throughout the first half of the Decade of the Kidney, we have raised awareness about the increasing burdens of kidney disease worldwide, among people, communities, workforces, and the governments. This effort has also accelerated a golden era of medicine and the determination to transcend status quo treatments, including brick-and-mortar dialysis, to a future kidney care that will be determined by expansions in patient consumer care choice, targeted therapies that stop kidney disease and prevent kidney failure, greater access to donated organs, new devices, and alternatives to human organs, including artificial wearable and implantable kidneys and xenotransplantation. We believe the time is now, and as we move through the next 5 years of this golden age of medicine for patients, care partners, clinicians, researchers, innovators, industry, and government leaders, to accelerate our bold actions to push forward modern diagnostics, devices, and therapeutics, and continue to remove outdated policies that block their adoption. No country is immune to the impacts of kidney disease, but the good news is every person watching has a role in shaping a future for all people who are at risk for or living with kidney disease that offers more hope, better outcomes, and greater independence and freedom from disease and disability. With that, I am honored to serve as the moderator for our session titled Hope Now for Rare Diseases, Accelerating the Path to Scientific Breakthroughs. Joining us, we have 3 esteemed speakers. Our first speaker is Dr. Sharon McGroder. Dr. McGroder is the Executive Director of the Rare Kidney Disease Foundation, an allied partner organization of AAKP's, founded by our good friend Richard Nelson, who was part of our inaugural Global Summit in 2019 to announce the stand-up of what has become a highly impactful and respected organization. Dr. McGroder spent 30 years as a family policy researcher in Washington, D.C., and now brings her skills and passion to raise awareness about autosomal dominant tubulointerstitial kidney diseases, ADTKD, a rare genetic condition that has affected multiple generations of her family, including herself. Now in stage 4 kidney disease and on the waiting list for the transplant, Sharon is committed to accelerating research and hope for others living with ADTKD. Please welcome Dr. Sharon McGroder.
Thanks, Suzanne, and I'd like to thank AAKP and George Washington University for sponsoring this event and asking me to talk with you all today about hope on the horizon for patients with ADTKD, which is autosomal dominant tubulointerstitial kidney disease. Today I'm going to talk about the numbers. What do we mean by ADTKD? How prevalent is it? Talk a little bit about ADTKD and how it's diagnosed, and talk about the importance of genetic testing. I'd also like to share with you breakthrough therapy that's on the horizon that's bringing hope to millions with this disease. And then I want to talk more generally about why it's important to invest in research on rare inherited diseases. I'll then share my personal story and talk a little bit about the Rare Kidney Disease Foundation. First of all, brief overview of the numbers. Up to 40% of dialysis patients don't know the cause of their chronic kidney disease. And up to 10% of all CKD patients in the US, and that's over 3.5 million people, have a genetic mutation that causes their kidney disease. ADTKD is the second most common genetic kidney disease after polycystic kidney disease, with an estimated 75,000 to 100,000 patients in the US alone and potentially millions worldwide. Here's a quote from Dr. Anna Greca, a partner of ours from the Broad Institute at MIT and Harvard. She says that rare genetic diseases are collectively common. And then Dr. Blyer, Anthony Blyer from Wake Forest University, another partner of ours, he makes the point that every nephrologist cares for at least one patient with ADTKD. So that speaks to the prevalence also of this otherwise rare disease. So what is ADTKD? As I mentioned, there's the three elements of it. Breaking it down, autosomal dominant means that parents have a 50-50 chance of passing it down to each of their children, and it does not skip generations. It is the dominant inheritance. It also means that if you are a CKD patient, chances are your parent also has this if it's ADTKD. So from both generations, there's a risk. Tubulointerstitial means that the disease occurs inside the kidney's tubules and interstitial cells, not in the glomeruli. And most kidney diseases occur in the glomeruli, but this one is different, which also kind of masks its presence because in the tubules, if that's where the disease is, you won't have blood or protein in the urine. It's what they call a bland urinary sediment. And this confuses a lot of nephrologists. If you don't have protein or blood in the urine, it does not come to them to think that it could still be a genetic kidney disease. And then the third element is that it's a kidney disease, but the key here is that it's slowly progressing over many, many years. And so you can look at the three red flags there. If you have those three red flags, there is a good chance you may have ADTKD. A little more detail about ADTKD. In this disease, gene mutations cause abnormal protein production that damages the kidneys, and that's what leads to the progressive decline in kidney functioning until transplantation or dialysis is required. There are 7 types of ADTKD that have been discovered so far, because the most recent one was just discovered last fall in October of 2024. So additional gene variants are being discovered all the time. Now, the 2 most prevalent types of ADTKD are what we call UMOD and MUC1, and they are so named because of the genes that are affected. In terms of diagnosing ADTKD, as I mentioned earlier, there are really no telltale signs, unfortunately. The kidneys are normal size, there are no cysts, there's no blood or protein in the urine, and kidney imaging and kidney biopsies do not help diagnose ADTKD. The really key red flag that can start the investigation into whether it could be this genetic disease is if there's a family history of chronic kidney disease and kidney failure with no known cause. And the most definitive test for ADTKD is genetic testing. So why might someone with chronic kidney disease, or for whom chronic kidney disease runs in the family, why might they consider genetic testing? First of all, if they want to provide an accurate diagnosis and avoiding what they call the diagnostic odyssey. This is when you bounce from doctor to doctor trying to figure out what's wrong, and nobody has quite all the pieces or the information they need for early detection and diagnosis of this disease. And unfortunately, sometimes those unnecessary visits lead to more invasive diagnostic procedures like a biopsy. Another reason is to help your nephrologist guide prognostic and therapeutic decision making. Another reason someone might want to consider genetic testing when they have a family history of chronic kidney disease is it could help them inform life decisions such as family planning, financial planning, the choice of health insurance, or even what job to take given the kinds of health insurance that might be offered or not offered. A few more reasons why you might want to consider genetic testing. If CKD, chronic kidney disease, runs in your family and you're considering being a kidney donor, you may want to rule out that you might have this genetic disease as well before you donate a kidney. If you want to screen at-risk but pre-symptomatic family members, if your kidneys seem to be functioning fine, but again, it runs in the family and you just want to know. And finally, you might want to participate in research to advance the development of a treatment. And I'll talk more about that in a couple minutes. And let me just note the source here for a lot of these reasons and discussions of genetic testing in the footnote there. So this is really exciting. There is a breakthrough therapy on the horizon because currently there are no treatments or cures for ADTKD. However, the brilliant physician scientists and cell biologists at the Broad Institute of MIT and Harvard have discovered a drug-like compound that shows promise for halting the progression of ADTKD. And this compound has been licensed to a pharmaceutical company with plans to conduct a clinical trial as early as 2027. So with that exciting news and hope on the horizon, I want to talk more generally now about why we should invest in studying rare inherited kidney diseases. First of all, it's amazing to learn that when you find out something about one genetic disease, it can help inform discoveries that benefit other diseases. For example, what Dr. Greca and her team at the Broad Institute discovered about ADTKD, they realized that the biological mechanism by which the ADTKD gene mutations lead to cell death and disease through these toxic proteinopathies, that's a common pathway for other diseases like ALS and Alzheimer's. And so even the treatment that they're discovering might help slow the progression for ADTKD. A similar treatment, now that they know the cause and the biological pathway, could show promise for treatments for these other diseases, like I said, Alzheimer's, ALS, and many others considered to be toxic proteinopathies. In fact, in her TED Talk, Dr. Greca estimates that more than 50 disorders could be amenable to this approach because of what they're learning about ADTKD. Second, an important reason to invest in rare inherited diseases, as I mentioned earlier, they may be individually rare, but they are collectively common. If 10% of Americans are suffering from a genetic disease, that's over 30 million individuals. Third, I think it's really important to emphasize that we in the rare inherited disease community, we really depend on investments in science to learn about this disease, to learn about these biological pathways that could lead to a treatment. And we really depend on investments by pharmaceutical companies who are willing to fund the trials, the clinical trials that are necessary to bring effective and safe medications to market. And it's also very important to invest in patient advocacy organizations like the Rare Kidney Disease Foundation, because what we do is critical. We raise awareness, we collaborate with the scientific community, and we build a supportive patient community for this disease, which, as I mentioned, is often invisible out in the community and even to nephrologists. So it's important that we all work together to bring awareness and help support scientists bring treatments to the market. In terms of my personal story, I grew up around kidney disease. My mother had it. My grandmother had it. My aunts and uncles had it. And it just was something that there was this unknown reason why many generations in my mom's side of the family died young from kidney disease. And my mom was one of 6, and 4 of them had kidney disease, and they all shared the same nephrologist who said that it wasn't genetic because he had diagnosed some with medullary cystic and some with polycystic. And because they thought these were completely different diseases, they thought it would not be genetic. And they didn't know at the time. This was back in the 70s and the 80s. So because of my family history, I started monitoring my kidney functioning when I was around 30 years old. And I was at stage 2. I was feeling fine. Life was good. And I had a nephrologist, so I was just going to keep an eye on it. And then in 2017, at the urging of a cousin who was diagnosed to her surprise with this disease, she found Dr. Anthony Blyer at Wake Forest University, who has been studying ADTKD since its discovery, since he discovered the first family back in 1996 in the Western Carolina mountains. And he is conducting breakthrough research on what we know about ADTKD. So I got in touch with Dr. Blyer. I got genetic testing through him and joined his study, and he confirmed that I have the MUC1 diagnosis or gene mutation of ADTKD. Currently, I'm at stage 4, waiting for a kidney donor, which fortunately my husband tested positive to be a donor. He's a match. And so I'm one of the fortunate ones who's been monitoring this over the years. And I've been preparing myself mentally, financially, and have had the blessing of my husband being a match when I need a kidney. So who is the Rare Kidney Disease Foundation, and how did I find them? I mentioned I joined the study with Dr. Blyer, and he's been a great advocate and a great supporter of kidney patients working together on behalf of our own needs. And I first heard of the Rare Kidney Disease Foundation a few years ago and joined as a volunteer about a year ago and was recently appointed its executive director. So the Rare Kidney Disease Foundation, or RKDF, we are a patient-driven, patient-focused nonprofit organization dedicated to halting the devastating impacts of ADTKD across generations by supporting physician scientists in their quest to find treatment and eventually a cure. Now, we are all volunteers, all of whom either have ADTKD or we have a family member with ADTKD. The Foundation's vision is that we see a world in which genetic kidney diseases are diagnosed early, treated effectively, and are eventually eradicated, thereby saving lives and rendering dialysis and transplantation unnecessary. And our mission is to offer hope to families with ADTKD, and we offer this hope by first raising awareness. What is it? What are the promising treatments on the horizon? And the importance of genetic testing. Second, we build engaged communities of medical professionals and patients and their families. And third, a primary goal is to find and refer patients to join the International ADTKD Registry at Wake Forest University, which will serve as a potential patient pool for the upcoming clinical trial. And we'd love for you to reach out to us. Here is our website, and you can contact us at info@rarekidney.org. And there are some QR codes for if you want some information for patients and families, information for physicians, information on genetic testing, and if you'd like to donate. So thank you, Suzanne, and thank you to AAKP and GW University, again, for sponsoring this global summit and for inviting me to speak to you today on this important topic.
Thank you, Dr. McGroder, for sharing both your personal journey and your powerful commitment to advancing research and awareness of rare kidney disease. Before we move on to the next speaker, I have a few questions for you. As an individual with a genetic condition, in your opinion, how can early diagnosis improve identification of rare kidney diseases, or help lead someone to consider genetic testing?
That's a great question. And let me first point out that chronic kidney disease itself is somewhat of a misnomer because there are so many different chronic kidney diseases that differ in the underlying cause. And unfortunately, too many CKD patients don't know why their kidneys are failing. They just know they have chronic kidney disease. That's why the sooner you can diagnose the underlying cause, the sooner the patient can receive personalized treatment tailored to their particular condition. For me personally, having seen so many family members across generations suffer and die from chronic kidney disease, I had simply assumed that it was genetic. So I began seeing a nephrologist, as I mentioned, at the age of 30, long before I was exhibiting symptoms. So just knowing I was at risk for kidney disease led me to monitor my kidney functioning from an early age. And I tried to adopt, healthy behaviors, see my nephrologist regularly. And then when it became necessary as my decline progressed, to go on cholesterol and high blood pressure medications. So I strongly advocate for early identification of chronic kidney disease through regular checkups, even with your primary care physician, and monitoring your blood work. Because again, at least for this disease and other kidney diseases, unfortunately, it's not until the creatinine starts to rise or the functioning starts to decline that signals an issue where you really should then get a nephrologist and start monitoring it over the years. So I guess it's when I found out that my kidney disease was genetic. So I was already in stage 4. So I had already been doing these preventative kinds of things. And by the time I knew it was genetic, I knew I was in serious decline. But for someone with a family history of chronic kidney disease, and they're pre-symptomatic, they may want to know they have a genetic kidney disease sooner than later so they can get out in front of it. So that's why we really kind of recommend that folks with chronic kidney disease who have a family history really talk to their nephrologist about genetic testing.
Through your work with Rare Kidney Disease Foundation, how are patients' experiences and perspectives helping to inform and elevate awareness and understanding of ADTKD among researchers and the broader kidney community?
Great question, Suzanne. We at the Rare Kidney Disease Foundation center the patient voice and their experiences in everything that we do. For example, we just had our 4th International ADTKD Summit over 2 days where we had experts talk about ADTKD, share their research on what they were learning. And in each of those days, we prominently featured patient voices and had them talk about their diagnostic journeys, how they finally got diagnosed, what it means to be living with this disease. And we thought it important for everybody attending and the scientific panelists and nephrologists in presence hear from patients and keep the patient voice centered in all that they do as well. We also connect interested patients with Dr. Bleier at Wake Forest University so they can obtain genetic testing if they would like and join the International Registry, which I mentioned is a group of individuals diagnosed with ADTKD through genetic testing. And they've agreed to participate in research so that the researchers can learn more about this insidious disease. And so by partnering with Tony Bleier and being sort of a conduit through which patients can get genetic testing, join these studies if they would like, and then once they're in the registry, that could be a potential patient pool for this clinical trial that they're hoping to start in the next 2 to 3 years. So we're really trying to bring the patients into the research aspect as well. And then a third major effort that we're doing, we're really excited about launching this year, an effort to elevate awareness of ADTKD is from the patient perspective, is what we're calling our Ambassador program. And we've already have 16 volunteers signed up to go out into their communities, starting with their own nephrologist and their own transplant centers, and hand out informational materials, flyers, describing what this is. Because again, many nephrologists are not aware of this disease, this genetic disease. And so just raising awareness one-on-one in person from a patient's perspective to say it's important that you start talking to your patients who have a family history of kidney disease about possible genetic testing. So we're really excited on the things that we're doing and coming up in the next few years where we're really engaging our volunteers, again, all of whom are patients or are family members of folks who have ADTKD.
And finally, in 2019, Richard Nelson, founder of the Rare Kidney Disease Foundation, was part of our inaugural Global Summit. Since that time, amazing strides have been made to stand up this organization as a beacon of hope and a community for those impacted by ADTKD. There have been many successes of this foundation over the past 5 years. What goals are priority for the foundation over the next 5 years?
Well, thank you for your kind words, Suzanne, about our successes. We're very proud of what we've been able to accomplish with our small but mighty team of dedicated volunteers. And as you know, Rich Nelson is a tireless advocate for ADTKD patients. So we're very honored to have him as our president and board chair. I would say we have at least 3 strategic priorities. for the next, I'd say, 3 to 5 years, certainly over the next 5, and hopefully we'll accomplish some of these even sooner. First, we want to raise awareness among nephrologists, as I mentioned, so that they're equipped to discuss ADTKD and genetic testing with their patients who have a family history of chronic kidney disease. And relatedly, we want to empower patients to talk to their nephrologists about genetic kidney disease and genetic testing if that's something that they want to pursue, and to empower them to feel confident in asking this of their nephrologists. Second, we want to help build the International ADTKD Registry, as I mentioned, at Wake Forest University, so that when the time comes for a clinical trial, there is a ready pool of eligible patients to draw from. And our third strategic priority, which will allow us to accomplish the first two and everything else that we do, is fundraising. If it weren't for the small donations of individual donors, most of whom are ADTK patients and their families, we wouldn't have been able to produce the materials and information that we have that we share with nephrologists. We wouldn't have been able to attend the American Society of Nephrologists last fall, and we plan to do every year, where we had a booth and handed out information and talked with these nephrologists, again, most of whom had never heard of ADTKD. We also hosted a panel reception and an expert panel discussion in the evening, where we, again, talked about this disease, the promising breakthroughs on the horizon. And for the 50 nephrologists in attendance, I think they learned a lot that they didn't know previously about this disease and why it's important for them to talk to their patients about it. We wouldn't, without these individual donors, have been able to mount 4 very successful international ADTKD summits. Our most recent one was about a month ago in early April. And that's what I mentioned, where it's a 2-day summit where we have experts present virtually. This was a virtual convening. And we had 200 attendees join us virtually to listen about the latest research, to get updates relevant to patients on how to manage this disease and how to talk to their nephrologists about genetic testing. And all of this was done through the small donations that we've received thus far from our small donors. And we wouldn't be able to partner with AAKP and George Washington and other partners to share information on websites and through webinars, educational webinars such as this one. So we're really grateful again to be able to share our message and our mission with your audience of kidney patients. And talk about the next 3 or 5 years, boy, with our ambitious ambassador program and all the educational materials and webinars and videos we want to produce. And we're going to need to obtain some grants that will allow us to sustain and effectively scale our efforts to help our research partners find a treatment and ultimately a cure for ADTKD.
Again, thank you, Dr. McGroder. We appreciate your leadership, partnership, and the work you're doing to drive progress for this often overlooked community. To close out today's program, we shift from the patient perspective to highlight the vital role of industry leadership in advancing solutions for rare kidney diseases. Our final 2 speakers represent industry partners who are deeply committed to this space and are at the forefront of efforts to drive innovation, improve outcomes, and support the global kidney community. It is with great pleasure that I introduce Dr. Daniel Jones. Dr. Jones serves as Vice President of U.S. Medical Affairs at Apellis Pharmaceuticals, where he and his team support launch efforts for specialty medicines in nephrology, hematology, and ophthalmology. Dr. Jones completed his PhD in neurosciences at UCSF, followed by a postdoctoral fellowship at Duke University Medical Center. Prior to joining Apellis, he had the opportunity to work on product launches across a range of neurology indications. Please join me in welcoming Dr. Jones.
Thank you so much, Suzanne. I'd also like to thank the AAKP and George Washington University for giving me and for Apellis Pharmaceuticals this exciting opportunity to talk a little bit about the work that we are doing in drug development, particularly in the nephrology space. First, I'd like to just share a little bit of important background information. This presentation contains scientific disease state information and is not intended to promote any Apellis product. This presentation isn't intended to be a substitute for consultation and review of reference materials and medical literature pertaining to individual clinical circumstances. Healthcare providers should make all treatment decisions based on the context of the situation and their clinical judgment. So at Apellis, we are very proud to have ushered in the first new class of complement medicine in about 15 years. And now we have 2 approved medicines that target C3, the central protein in the complement cascade. We delivered the first ever treatment for geographic atrophy, a leading cause of blindness worldwide, and a complex disease that researchers in the field had spent decades working to solve. We also brought forward the first approved targeted C3 therapy with the goal of elevating care for people living with a rare and potentially life-threatening blood disease. And at Apellis, we really believe that we've only just begun to scratch the surface and unlock the potential of targeting C3 across many serious diseases. And we're thrilled now to be over 700 employees globally. And I'll be talking a little bit more about the pipeline and why we're so excited about it. So at Apellis, as I mentioned, we really believe that we've just only begun to unlock the potential of targeting C3. And what you can see here in our pipeline is that we're focused on 3 key areas: ophthalmology, specifically retina, rare disease, which is where I'll be spending most of my time today, and neurology. And then earlier in the pipeline, a few additional exciting areas focused largely on gene therapy. Our cutting edge research is really focused across these 3 key areas. In ophthalmology, we are excited to have Syfovre approved for geographic atrophy secondary to age-related macular degeneration. We're also exploring other ways to further advance the care and treatment of geographic atrophy. In rare disease, we have Empaveli approved for PNH, but we're also studying this product in multiple kidney diseases, including C3G and IC-MPGN, where we have recently completed phase 3 trials and we have filed for approval with the FDA with an expected decision date later this summer. In addition, we are launching clinical studies in FSGS and DGF to additional kidney diseases with significant unmet need. So, why are we so interested in the complement system and specifically C3 as a target? The immune system protects the body by finding and fighting infections, removing harmful substances, and helping with healing. The complement system, where we focus our drug development efforts at Apellis, is a key part of this defense that helps immune cells by marking invaders, causing swelling to fight infection, and directly killing harmful microbes. Complement component 3, or C3, is a protein that helps your body fight infection and remove damaged cells. When C3 is overactive, it can lead to inflammation and kidney damage. C3 communicates with other proteins in the complement cascade that cause inflammation, like C3a and C5a, cell removal, like C3b, and lastly, cell death, like C5b. All of these collectively contribute to kidney damage. And our research at Apellis revolves around targeting these critical components of the complement cascade to ameliorate diseases that occur as a result of overactivation of this all-important system. But we think that the promise of targeting the complement system does not end there with kidney disease. Complement overactivation underlies many serious diseases, and of course, where we're focused mostly is on hematology, on kidney, and on ophthalmology, but we could not be more excited about the potential of targeting complement to have an impact on many, many diseases with unmet need. So with that, again, I'd like to thank Suzanne, the AAKP, and George Washington University for this opportunity to speak a little bit about our work in the nephrology space at Apellis. And with that, I'd be delighted to take any questions. Thank you.
Thank you, Dr. Jones, for your presentation. I have a few questions I'd like to ask you before we end. Dr. Jones, you shared how Apellis is approaching innovation in rare kidney diseases. Can you speak more about the role of patient-centered research in guiding your development of new therapies, and how patient input shapes clinical trial design and outcome measures?
That's a terrific question. At Apellis, patient-focused drug development is really a key pillar of how we conduct our research and development efforts. We work closely with kidney disease advocacy groups and individual patients and caregivers to make sure that we understand the ongoing unmet needs that face the patient community and what barriers they may be facing as they consider participating in clinical research. We bring patients and caregivers together to provide us with feedback regarding how we design our studies. This includes asking patients to tell us if the study endpoints that we're looking at are in line with how they measure and think about their own disease progression. We ask how we can make it easier for study participants to remain in our studies by looking at our assessment schedules and how we're providing them support. And we ask if our eligibility criteria accurately represents the patient population, and if there are any barriers that we can help to remove that might have the unfortunate consequence of excluding large numbers of patients from participating in our clinical research. At Apellis, we always say that what we do for patients, we do with patients, and this very, very much includes all of our clinical research efforts.
For patients or families affected by rare kidney diseases, what advice do you have for staying engaged in research and learning about potential new treatments that may not yet be made widely available?
Yeah, I think one of the most important things that patients and caregivers can do is to make sure to speak to your nephrology care team, first and foremost, and really specifically ask them about ongoing clinical trials that are in the space of the kidney disease that really matters to you. For so many rare kidney diseases, there are no approved treatments, unfortunately. So for some patients, a clinical trial may very well be the best option to gaining access to the latest and most innovative approaches in nephrology. Apellis has always worked with our advocacy partners on creating awareness around our clinical research. And we believe that by working together, we've got the best possible opportunity to deliver innovation and advance the kidney field forward in service of better patient outcomes.
And finally, Dr. Jones, as we reach the midpoint of the decade of the kidney, how can patients, particularly those impacted by rare kidney diseases, play a meaningful role in ensuring that research translates into real-world impact, including changes to treatment, guidelines, improved access to care, and decisions made by federal agencies and policymakers?
Well, there's no question that patients play an absolutely pivotal role in the development of new medicines, whether in the kidney space or beyond. So at Apellis, we really encourage patients to get involved with advocacy organizations that work on policy initiatives and that speak to decision makers to help them understand what it's like to live with a rare and chronic kidney disease. It is so impactful when patients are able to tell their own story and affect change, whether it's helping those on Capitol Hill understand barriers that patients may face to accessing new treatments, or letting them know what's most important to the patient community so new medicines can be developed ultimately with the patient's needs in mind.
Again, thank you, Dr. Jones, for sharing your insights and the important work Apellis is doing to support patients living with rare diseases. Moving on, it is my pleasure to introduce our final speaker for today's session, Dr. David Fuller, Chief Medical Officer for Dimerix. Dr. Fuller is an internationally experienced pharmaceutical executive and physician with over 35 years of international experience in drug development and commercialization. He has designed and executed multiple GCP Phase 1 to 3 studies across the U.S., Europe, and Asia for both orphan and major markets. At Dimerix, Dr. Fuller, responsible for is responsible for the strategic delivery of the company's flagship Action 3 Phase 3 clinical program for focal segmental glomerulosclerosis kidney disease, which he joins us today to speak to. Dr. Fuller, I turn things over to you.
Well, good morning, everyone. It's a pleasure to be here, and I'd like to thank Suzanne, AAKP, and the George Washington University for making this possible. My name is David Fuller. I'm the Chief Medical Officer of Dimerix, and today I'm delighted to be talking to you about our Phase 3 study in patients with FSGS called Action 3. Before we get there, though, we're going to talk about some fun facts about Australia. So let's start out here. Which of the following is not an Australian animal? Now, two of those should be very obvious, and leaving just maybe two to choose from. And the answer is number A. That bird there is the kiwi, which is a very cute resident of our neighbouring island, New Zealand. Moving on, does anyone know which country is bigger, Australia versus the USA? Lots of options there, including a reference to the dangerous animals we have living on and around Australia. So, of course, the USA is bigger than Australia, just slightly. However, we are the world's smallest continent, but largest island. And you might be interested to know that we only have 8 states and territories in that large size. That's because most of the country in the middle there is desert and uninhabitable. Similarly, we only have roughly a tenth of the population of the US. We only have 26 million, again, because much of Australia is uninhabitable. We simply don't have enough fresh water in the middle of the country. But despite all those venomous animals you saw on the previous slide, despite that, we still rate, 2 of our cities rate as the most livable cities in the world, Melbourne and Sydney. So some facts about Dimerix and who we are. We are a small Australian biotech headquartered in Melbourne, Australia. We are publicly traded on the Australian Stock Exchange. The ticker code is DXB if you're interested. We are small, just 15 employees, but we do a lot with those 15, and we are currently running the largest Phase 3 trial for FSGS patients in the world, and we hope that trial will bring a new treatment to patients very soon. So let's move on now to talk about the trial, the product, how it works, and what we've done so far. You'll know that FSGS is a podocytopathy, and I'm going to show you a picture of that on the next slide. FSGS is an area of high unmet need in both adults and children, and sadly, it typically progresses to end-stage renal disease in roughly 7 to 8 years, so quite quickly from diagnosis. There are no current approved FDA treatments. Treatment is typically empiric and consists of high-dose steroids or non-steroid immunosuppressants like tacrolimus or cyclosporin, which, if you've taken them, you'll know are associated with some significant side effects. So new treatments are desperately needed. So you can see here a picture's worth 1,000 words. On the left is a normal podocyte, and you can see there the cartoon of the damage in the podocyte. And what that damage does is it allows the kidney to leak protein. In normal kidneys, protein is not excreted. It's prevented from leaving by the filtration barrier, which the podocytes create. So when the podocytes are damaged, that damages the filtration barrier and means that protein appears in the urine. And we can see that on the next slide. This is a wonderful picture from an article by Hans de Vries. And you can see there on the left, slide A is a normal podocyte looking very pretty. And then you can see as we move to B and C, the increasing damage, which looks not particularly good. So that's basically FSGS in pictures. And so what we know about glomerular disease is that once damage occurs, we enter this vicious cycle of damage whereby the initial damage, whether that be inflammation or something else, causes this damage to the podocytes that often leads to hyperfiltration and hypertension, renal fibrosis, proteinuria, and loss of nephrons. So you get increasing damage leading to end-stage renal disease. So how do we break that cycle? Well, we believe the answer is the pharmacologic rationale that we have in Action 3. So we target two receptors. One is CCR2, the chemokine receptor 2, and the other is the angiotensin type 1 receptor. Both of these have been independently implicated in the initiation progression of fibrosis in a range of diseases. Now, CCR2 is required for recruitment of inflammatory cells to the kidney, and AT1R antagonism has been shown to have a kidney protective effect. So we hypothesized that combining those two receptors, inhibition of those two receptors would be a very good thing. And certainly CCR2 has been shown into mouse disease models to, if we inhibit CCR2, we decrease proteinuria and reduce glomerular injury. And importantly, we know that glomerular expression of both CCR2 and the ligand for that receptor, which is called MCP1, is significantly increased in human FSGS biopsies. So we know CCR2 definitely plays a role in FSGS, and that by inhibiting CCR2 as well as the AT1 receptor, we believe it's a case of 1 + 1 = 3. And I can show you some data we have that supports that hypothesis. So we conducted a test in animals in the so-called subtotal nephrectomy model, where we looked at DMX-200 alone, the AT1R inhibitor alone, plus then the combination, which is the purple box at the bottom. And if we click forward on the slides, we'll see the results. And you can see there that in all three cases, starting from the top, the combination in purple preserved podocytes much better than either agent alone. We also reduced macrophage infiltration much more strongly in combination. And most importantly, you can see at the bottom that the combination of both agents reduces proteinuria. So I think you'll agree that's a very robust demonstration of the action, of the pharmacological action that underpins Action 3. And now on this slide, this shows how the various touch points that both DMX-200 and the angiotensin blocker play in interrupting that cycle of damage. So you can see there's quite a large impact in modulating that cycle of damage. So that's why we're very quietly confident about the product. This slide here shows our total clinical development experience, starting on the left with a phase 1 study in healthy volunteers. And then we conducted 3 small phase 2 studies, the first one being a dose-finding study, the second being a very small study in FSGS 8 patients, the third being a 45-patient study in DKD, and finally the commencement of Action 3 on the right there. Importantly, we saw positive efficacy signals, reductions in proteinuria in all studies. We selected the dose of 120 milligrams twice daily, and we saw very few adverse events. And in fact, the adverse events rates in those phase 2 studies were very similar between the active and placebo arms. And we'll talk more about that later in the presentation. So this slide here shows the proteinuria results of our small phase 2 study. Now, being only 8 patients, we're not going to see statistical significance, but you do see there that the combination has an increased drop in proteinuria compared to placebo. And then if we click forward, we'll then see the results of the biomarker, the MCP1, which binds to the CCR2 receptor. So what this is showing is that the combination of DMX-200 plus the ARB is resulting in a reduction in MCP1. So the CCR2 inhibition is working, which is exactly what we want. So this data essentially gave us the confidence to proceed into a larger study, the phase 3 Action 3 trial in FSGS. And we'll talk about that now. So let's talk about the Action 3 study at a high level. The study targets are we would like to recruit 286 adult patients with FSGS by Q4 this year. And in addition, we also have a requirement to recruit 22 adolescents, and that will occur by the end of next year, 2026. Currently, I'm very pleased to tell you that we've had a flurry of activity and we're now under 100 patients to randomise, and we're aiming for a total of 50 randomisations from the US. So for those who might be interested in participating, there's plenty of time to be involved, and we are looking for more patients. Importantly, the study has passed a formal futility assessment, which means we're confident that something is happening. And it's also passed an additional 4 IDMC reviews. That's an independent data monitoring committee. And each time the IDMC has reviewed the study, they have made no changes to the trial, and they have not identified any safety issues of concern. So we are quietly encouraged by the progress of the study so far. In terms of our rare disease community partners, we rely heavily on our partners, including the Neptune group, NephCure, who are the rare disease kidney specialists. Obviously, we're working with AAKP. And then we have 2 individuals, our patient advocate, Curtis Warfield, and also Kevin Fowler, Voice of the Patient. So very much conscious that in rare disease, it's very important, even more important than normal, to be working directly with patient groups. This is a brief summary of where we're working. You can see that we're working in 21 countries in over 200 sites. As I mentioned, in the US, we'll have about 50 sites. Here's a summary of our slides spread across the US. So I'm very pleased that we've got quite broad geographic spread. Some sites are adult only, some sites are pediatric only, and some sites do both. At the end of the presentation, I've got a link to the study website where you can see some of this in more detail. Here's a summary of recruitment to date. You can see here that recruitment has been relatively evenly spread across the world. The US is not far from being the top recruiter. It does screen a lot of patients, but unfortunately quite often the patients are screened failed, typically because there's not enough proteinuria or the EGFR is too low, and occasionally because the patient has secondary FSGS, which we exclude, and I'll talk about that coming up. So let's move on to talk about the protocol in more detail. So the Action 3 study is a pivotal randomized double-blind study to investigate the efficacy and safety of DMX 200 compared with placebo over a period of 104 weeks or 2 years in both adult and adolescent patients with FSGS. The study has a 1-to-1 randomization, which means patients have a 50% chance of being allocated to active. And the study requires a total of 13 visits over 2 years and 1 month. So that means on average, you're visiting the study site every 2 months. So it's not too heavy on visits, and we've worked hard to try and reduce the burden on the patients. The dose is 120 milligram capsule taken orally twice a day, morning and evening. And importantly, all patients who complete the double-blind period qualify to participate in an open-label extension where everyone will receive DMX 200. And we've had a very high level of conversion to the open-label extension. We have over, I think, 95% of our patients have chosen to convert. So I think that's a very positive sign. So here is the study outline in concept, and you can see we've got various periods. So there's an initial screening period to make sure patients are eligible to participate. And then if patients are not on an ARB, they have the opportunity to transition onto an ARB. Typically, they will be on an ACE inhibitor, and then we just switch the ACE inhibitor for an ARB inhibitor during that 4-week titration period. It's up to 4 weeks. It can sometimes just take 1 day to titrate, so that's quite variable. And then the important part is everyone then enters a 6-week stabilization period. That's to wash out so-called hemodynamic changes, just to make sure any changes results in a stable baseline for us. And 1 week before the end of the stabilization period, there's a qualification visit where we recheck your proteinuria to make sure you still qualify. And then assuming you qualify, you're then randomized on a 1-to-1 basis and enter the 104-week double-blind treatment period. At the end of the study, there's then 4 weeks off therapy. Why do we do that? It's so we can assess duration of effect and carryover effect. That will give us some quite important information. And then after that 4 weeks off therapy, people can enter the open label extension component of the study. And if we click forward one slide, we can see the visit schedule. So there you can see 13 visits over that 108-week period. Of course, most of the visits are sort of front-end loaded. So there's 6 visits in the first couple of months, and then the visit schedules become lengthier. Okay, let's talk about our key inclusion criteria. We have a very wide age inclusion from 12 to 80 years old. In terms of FSGS, we accept 3 of the primary FSGS types: primary genetic or undetermined cause FSGS, diagnosed within 7 years of screening. The restriction to 7 years is to reduce variability in the trial, and it's not a prejudice for those who've have FSGS longer. We're just trying to make sure that this trial is successful. The other thing to note is we do not accept secondary FSGS. Secondary FSGS is typically due to another disease, for example, diabetes or hepatitis C, and so we need to exclude secondary FSGS. In terms of eGFR, we accept eGFR between 25 and 120 mils per minute. And for proteinuria, there are 2 ways to come in: either your urine protein creatinine ratio being greater than 1.5 gram per gram, or you can come in if your 24-hour urinary protein is greater than 1.5 grams a day. So we sort of give you 2 shots to come in on proteinuria. We allow people with a BMI with 40 or less to come in, so that's quite a wide range there. And of course, as we mentioned, you do need to be on an ARB, an angiotensin receptor blocker. That's at least 50% of the labeled dose for hypertension. And as we discussed on an earlier slide, we build in a period in the trial where you can convert to an ARB if you're not on one already. We do allow low-dose corticosteroids, 10 milligrams or less per day. And if you're taking other things like aldosterone inhibitors or direct renin inhibitors or SGLT2 inhibitors, we just ask that the dose and regimen of those is stable for 12 weeks prior to starting the trial. And blood pressure there you can see has to be less than 160 on 100. Let's talk about some of the key exclusions now. So key exclusions, as we talked about previously, FSGS, which is secondary to another condition. Also, if patients have nephrotic syndrome, which is very high proteinuria combined with a low serum albumin, patients with that should have been treated with standard of care first. So patients with nephrotic syndrome must receive standard of care treatment first before they can become eligible for this trial. We have to exclude a history of type 1 diabetes, but if you have controlled type 2 diabetes, you can come in. Uncontrolled type 2 diabetes cannot come in. And that's because of the high risk of secondary FSGS in that population. No prior organ transplants, including kidney transplants. Serum potassium must be less than 5.5 for safety reasons. And probably the exclusion that impacts most patients is the fact that you cannot be on non-steroid immunosuppressant drugs like tacrolimus or cyclosporin. If you have been treated with those agents, we ask that you stop them for 12 weeks prior to being screened for the study. That's because those agents impact the same pharmacological pathway that we target, so we have to do that for signal detection purposes. So what happens if you need additional medication during the trial? The short answer is you can have it, we term it rescue therapy. So if for some reason your FSGS worsens during the trial, you can be treated with whatever treatment your doctor feels is necessary, and you can continue on in the trial provided there are no safety issues. We now know the trial's been running a very long time, that we don't appear to have any significant drug interactions. On this slide, there's just a brief overview of the open label extension. So once you complete the double-blind period, all patients qualify to go into the open label extension, where everyone will be receiving DMX 200. And you can see that there are very few visits in the open label extension, so it does not demand too much time or effort from a patient perspective. So the trial has been running for some time now, so I thought you'd like to just have a summary of what we know about what's happening in the trial so far. So I can tell you that the average patient has now received over a year of therapy with DMX 200 or placebo. And that importantly, the study has passed a formal futility assessment, which means that there is something going on in terms of reduction in proteinuria. I don't know how much, but we know that obviously there was something happening. The study's also received 5 further IDMC reviews, and we've had no changes to the study and no safety issues have been identified. If I summarise the safety data into a simple to understand terminology, the average patient on our study experiences 3 to 4 mild to moderate adverse events per year. And if you think about that, certainly from my perspective, definitely I would experience a couple of headaches a year, probably have a cold and the flu. So as you can see, it's not a high level of adverse events. And what I say is that I'm confident that this product does not add a burden of side effect to patients, which I think is what we need in FSGS. The current major treatments, high-dose steroids and immunosuppressives, do have some serious side effects. So I'm very pleased to tell you that the agent does appear to be very well tolerated and suitable for chronic use, which I think is very important for FSGS. So I'm sure you'd like to know more about the trial. If so, please feel free to visit our study website shown there. And also, I'm very happy to be contacted directly. My email address is there for any queries. The study website will show you your closest open site, so it is a source of very good information. And with that, I'd just like to thank AAKP again for the great opportunity to present the slide. I hope you have a great meeting, and we really appreciate the opportunity to participate.
Thank you, Dr. Fuller, for your presentation. I have a couple questions to ask you. Dr. Fuller, what are the key challenges you face in the clinical development of therapies for rare kidney diseases, and how can patients, care partners, and healthcare providers help address these barriers?
Wow, what a great question. And I'll answer that in a number of ways. There are several challenges. I think the biggest challenge in rare disease is that patients often don't know about the clinical trial. That's probably our biggest barrier. We know that probably less than 1% of eligible patients actually participate in rare disease renal studies. So that's a significant barrier. So the work of organisations like AAKP and NEFCURE is invaluable in helping in that regard. The other barriers in rare disease are the fact it is rare, and so it's hard to find patients, and often the patients are spread out. So in the case of our trial, we've had to recruit 286 patients. We've had to open over 200 sites. So what that means is most sites will only recruit 1 patient. So the trial is not very efficient from that perspective. And basically what that means is the trial is quite expensive to run. And so that's I guess a commercial barrier for a lot of companies. They're probably the 2 biggest challenges I can think of.
As we reach the midpoint of the decade of the kidney, how can patients play a meaningful role in ensuring that research in the rare kidney disease space translates into real-world impact, including changes to treatment guidelines, improved access to care, and informed decisions by federal agencies and policymakers?
Well, that's another great question. I think a short answer is for patients to speak up and get involved. Ask your nephrologist, do you do clinical trials? Clinical trials are the main way that we progress innovation. If they're not participating in clinical trials, ask them why not. And then the other important thing is for the patient voice to be heard. So the only way the patient voice can be heard is to be active in the community. So I'd encourage everyone to support organisations like AAKP, who are essentially the voice of the patient. The more people involved, the more noise we make and the more progress we'll make.
Thank you very much for being with us today, Dr. Fuller. As we wrap up today's session, I would like to extend a sincere thank you to all our speakers, Dr. Sharon McGroder, Dr. Daniel Jones, and Dr. David Fuller, for sharing their invaluable insights into the world of rare kidney diseases and providing an overview of advancements in research and treatment that are occurring in this space.